Bismillah ar-Rahman ar-Rahim. This is Ahtar Totahil, Al-Farabi University Faculty of Medicine students, second year students. I want to present my presentation on hormone synthesis and transport to their target organs. So now, before that, we explain what is hormone and which cells are targeted by this hormone. First, we will describe that the multiple activities of the cells, tissue, and organs of the body are coordinated by their interplay of several types of chemical messenger systems. First of them is neurotransmitters released by axon terminals of neurons into the synaptic junctions and acts locally to control nerve cell functions. Second, endocrine hormones released by glands or specialized cell into the circulating blood and influence the function of cells at another location in the body. Third one is neuroendocrine hormone secreted by the neurons into the circulatory blood and influence the function of the cells at another location in the body. Four is paracrines secreted by cells into the extracellular fluid and affect neighboring cells of different types. Five, autocrines secreted by cells into the extracellular fluid and affect the function of the same cells that produced them by binding to cell surface receptors. 6. Cytokines. Peptides secreted by cells into the extracellular fluid and can function as autocrines, paracrines, or endocrines hormones. Examples of cytokines include the interleukin and other lymphokines that are secreted by helper cells and acts on the other cells of the immune system. These are autocrines, these are paracrines as I explained, and these were endocrines. What are hormones? Natural organic substance that regulate growth, metabolism, and other function of an organism. They are simply biochemical messengers. They can be classified according to their chemical composition organs, where they work, for example, reproductive hormones in the reproductive organs, and if they either act on the same cells producing them, for example, autocrine or paracrines. And the multiple hormone system play a key role in regulating almost all body functions, including metabolism, growth and development, water and electrolyte balance, reproduction and behavior. Chemical structure and synthesis of hormones. There are three general classes of hormones. First is protein and polypeptides, including hormones secreted by the anterior and posterior pituitary gland, the pancreas, insulin, and glucagon, which is secreted by beta Langerhans cells, insulin, the parathyroid gland, parathyroid hormone, and many others. The second one is steroids secreted by the adrenal cortex, cortisol and aldosterone, the ovaries estrogen and progesterone, the testes testosterone, and the placenta estrogen and progesterone. 3. Derivatives of the amino acid tyrosine secreted by the tyrosine thyroxine, and triiodothyronine, amines T3 and T4, and the adrenal medulla, epinephrine and norepinephrine. There are no known polysaccharides or nucleic acid hormones. Polypeptide and protein hormone are stored in secretory vesicles until needed. Most of the hormone in the body are polypeptides and proteins. In general, polypeptides with 100 or more amino acids are called protein, and those with fewer than 100 amino acids are referred to as peptides. Synthesized on the rough end of the endoplasmic reticulum of the different endocrine cells. Uh, they Now we want to discuss that they are usually synthesized first as larger proteins that are not biologically active. Pre-prohormones and are cleaved to form smaller prohormones in the endoplasmic reticulum. These are then transferred to the Golgi apparatus for packing into secretory vesicles. In this process, enzymes in the vesicles cleaves the prohormones to produce smaller biologically active hormones and inactive fragments. The vesicles are stored within the cytoplasm and many are bound to the cell's membrane. Their secretion is needed. Secretion of the hormone as well as inactive fragments occur when the secretory vesicle fuse with the cell membrane and the granular contents are extruded into the intestinal fluid or directly into the bloodstream by exocytosis. The stimulus of exocytosis is an increased in cytosolic calcium concentration caused by depolarization of the plasma membrane in other two instances stimulation of an endocrine cell's surface receptor. The peptide hormone are water soluble, allowing them to enter to circulatory system easily, where they are carried to their target tissues. Storage. Polypeptide and protein hormones are stored in a secretory vesicle until needed. Most of these hormones in the body are polypeptide and proteins. As I mentioned before, that these polypeptides with 100 or more amino acids are called protein, and a fewer than 100 amino acids are peptides, synthesized on the rough end of the endoplasmic reticulum. Steroid hormones are usually synthesized from the cholesterol and are not stored. The chemical structure of the steroid hormone is similar to that of cholesterol, and in most instances they are synthesized from cholesterol itself. Consist of three cyclohexyl rings and one of cyclopentyl ring combined into single structure. Because of these steroids are highly lipid soluble, once they are synthesized, they simply diffuse across the cell membrane and enter the interstitial fluid and then to the blood. Amine hormone are derived from tyrosine. The two groups of hormone derived from tyrosine, the thyroid and the adrenal medullary hormones, are formed by the action of enzymes in the cytoplasmic compartment of the glandular cells. Control of hormone secretion. Most hormonal regulatory systems work via negative feedback, but a few operate via positive feedback. Negative feedback prevents overactivity of hormone system. The control variable is often not the secretory rate of the hormone itself, but the degree of activity of the target tissue. Therefore, only when the target tissue activity rises to an appropriate level will feedback signal to the endocrine gland become powerful enough to slow further secretion of the hormone. Feedback regulation of hormones can occur at all levels, including gene transcription and translation steps involved in the synthesis of hormones and steps involved in processing hormones or releasing stored hormones. Cyclical variation occur in hormone release superimposed on a negative and positive feedback control of hormone secretion or periodic variation in hormone released that are influenced by seasonal change, various stage of the development and aging, the diurnal daily cycle, and sleep. For example, the secretion of growth hormone is markedly increased during the early period of sleep but reduced during the later stage of sleep. Transport of hormone in the blood. Water-soluble hormone, peptide, and catecholamines are dissolved in the plasma and transported from their site of synthesis to target tissues, where they diffuse out of the capillaries into the intestinal fluid and ultimately to the target cells. Steroid and thyroid hormones, in contrast, to circulate in the blood mainly about bound to plasma proteins, usually less than 10% of the steroid or thyroid hormones in the plasma exist free in solution. However, protein-bound hormones cannot easily diffuse across the capillaries and gain access to their target cells, and therefore biologically inactive until they dissociate from the plasma proteins. The relatively large amount of hormones bound to the proteins serve as reservoirs, replenishing the concentration of the free hormones when they are bound to the target receptors or lost from the circulation. Binding a hormone to plasma protein greatly slow their clearance from the plasma. They distinguish between hydrophilic and lipophilic proteins, peptides, hormone, and catecholamines, and lipophilic steroid and thyroid hormones. Primary act through second messenger system and activate gene and binding with the receptors in the nucleus. Circulate mainly dissolved in the plasma, largely bound to the plasma protein. Clearance of hormone from the blood. Two factors can increase the or decrease the concentration of hormone in the blood. A. The rate of hormone secretion into the blood. B. The rate of removal of hormone from the blood, which is called metabolic clearance rate. This is the usually expressed in term of number of milliliter of plasma clearance in the hormone per minute. To calculate this clearance rate on the measures, first the rate of disappearance of the hormone from the plasma per minute, and the second the concentration of the hormone in which the milliliter of plasma. Then the metabolic clearance rate is calculated by the following formula. Metabolic clearance rate is equal to rate of disappearance of the hormone from the plasma divided by concentration of hormone in each milliliter of plasma. Hormones are cleared from plasma in several ways, including metabolic destruction by the tissues, and the second, binding with the tissues. Third is excretion by the liver into the bile, and fourth, excretion by the kidney into the urine. For certain hormones, a decreased metabolic clearance rate may cause an excessively high concentration of a hormone in the circulatory body fluid. For instance, this occurs for several of the steroid hormones when the liver is diseased because their hormones are conjugated mainly in the liver and then cleared into the bile. Uh, Hormones are sometimes degraded in the target cell uh, by enzyme processed that cause endocytosis of the cell's membrane hormone receptor complex. The hormone is then metabolized in the cell, and the receptor are usually recycled back to the cell membrane. Most of the peptide hormone and catecholamines are water soluble and circulate freely in the blood. They are usually degraded by the enzyme in the blood and tissue and rapidly excreted by the kidney and liver, thus remaining in the blood for only a short time. Hormones that are bound to plasma protein are cleared from the blood at much slower rate and may remain in the circulation for several hours or days. General um, principle of a hormone action. General principle of hormone action. Uh, a tropic hormone, a hormone that has its primary function, the regulation of hormones secreted by another endocrine gland. Synergism, when different hormones work together and have a greater effect than individual hormone action. And the uh, third one, sorry, screen sharing. The second one is synergism. When different hormones work together and have a greater effect than individual hormone action. The third one is permissiveness. Uh, a small amount of one hormone allows a second hormone to have its full effect on a target cell. For example, first hormone permits the full action of the second hormone. Uh, Antagonism. One hormone produces the opposite effect of the other. The process involved in both the negative and positive feedback. For example, if A greater than B and B is greater than C and C is greater than D, increase in D causes inhibition of A. For example, negative feedback, if D decreases A, produce is triggered. For example, positive feedback. So in this schema, we see that the plasma concentration of free biologically active hormone, which can be interact with its target cells to produce a physiological response, depend on the first number is secretion, the hormone's role of the secretion by the endocrine gland for a hormone. The major factor, it's the major factor that from endocrine gland. Then to the plasma hormone bond to the plasma protein, and this activity happens. Second, activation of some hormones. Its rate of metabolic activation for a few hormones. Then in plasma, binding lipoic hormones from free biologically active hormones. Its extent of binding to plasma protein for lipoic hormones, and four number for then these go to metabolism in liver or other tissues and or it go to target cells or physiological response. When they go to metabolic in the liver and other tissues, the rate of inactivation, metabolic inactivation and exertion for the hormone is dependent on its excretion in urine. Hormone receptors activation and their activation. The first step of the hormone's action is to bind to a specific receptor at the target cell. The location for this different type of hormone receptor are generally the following. First, in or on the surface of the cell membrane. The membrane receptor are specific mostly for the protein, peptide, and catecholamine hormones. And the second, in the cell cytoplasm, the primary receptor for the different steroid hormones are found mainly in the cytoplasm. The third number is in the cell nucleus. The receptor for the thyroid hormone are found in the nucleus and are believed to be located in a direct association with one or more of the chromosomes. Here we see that receptors for the target hormone targeted, they are internal and external. What it means? Internally, that they are existed in the cytoplasm and nucleus, and externally that are located in the cell membrane. Dear students, as we are in contact, now we will discuss that how lipid soluble hormone works. Uh, Binding to specific cell receptor in the cell membrane and form membrane cell receptor uh, complex which diffuse to nucleus. The receptor is eventually released from reuse. Steroid activates a specific gene to produce messenger RNA. Messenger RNA pass out into the cytoplasm and initiate protein enzyme synthesis, uh, and a free lipid soluble hormone molecule diffuse from the blood through interstitial fluid and through lipid bilayer of the plasma membrane into the cell target cells. The hormone bind to and activate receptors located within the cytosol or nucleus. The activated receptors hormone complex then alter gene expression, turns specific gene of the nuclear DNA on or off. As the DNA is transcribed, a new messenger RNA forms, leave the nucleus and enter the cytosol, then a direct synthesis of new protein, often an enzyme or the ribosomes. The new protein alter the cell's activity and cause the response typical of hormones. Now, the question arises that why do they penetrate the cells? The response is that the whole process is called mobile receptor hypothesis, in which a steroid hormone is not attached to the plasma membrane but seem to move freely in the nucleoplasm. And the steps for the lipid soluble hormones, how it works. The step 1, free lipophilic hormone not bound with the plasma protein carrier, diffuse through the plasma membrane of the target cell and bind with the receptor, which is intracellularly located inside the cytosol or in the nucleus. Step 2, each receptor has a specific binding region with the hormone and another region with binding with DNA. Receptor alone cannot bind to DNA unless it binds to hormone. When the hormone is bound to receptor, the hormone receptor complex binds to specific region of DNA known as hormone response element. Step 3 is transcription of gene. Step 4, messenger RNA transported out of nucleus into the cytoplasm. Step 5, translation at ribosome. Step 6, protein enzyme released from ribosome. Step 7, protein enzyme mediated ultimate responses. Now, in this schema, we see that uh, this is blood vessels and then protein carrier. This is a steroid hormone. The most hydrophilic steroid are bound to plasma protein carriers. Only unbound hormone are diffused into the target cells. Then number 2, this to the cytoplasmic receptor, steroid hormone receptor are in the cytoplasm or nucleus, And the three which go to here, the receptor hormone complex bind to DNA and activate or repress one or more genes. And four, uh, for the translation, activated gene create a new messenger RNA that move back to the cytoplasm. And five, to the new protein translation produce new protein to cell process. And six, repeat responses to cellular surface receptor. Some steroid hormone also bind to membrane receptor that are a second messenger system to create lipid cellular response. Uh, Hydrophilic hormones get transported to their target site freely in the plasma since they are soluble in it. On getting to the target cells, they are unable to pass through the plasma membrane because of their hydrophilic nature, which contrast with the hydrophobic nature of the membrane interior. As a result of this, there is need for second messenger that will relate the message carried by the hormone to the cell. How water soluble hormones works. Uh, and a water soluble hormone, the first messenger diffuse from the blood through interstitial fluid and then bind to its receptor at the exterior surface of target cell's plasma membrane. The hormone receptor complex activate a membrane protein called G protein. The activated G protein in the turn activate adenylate cyclase. Then adenylate cyclase convert ATP into cyclic AMP because the enzyme active site is on the inner surface of the plasma membrane. This reaction occurs in the cytosol of the cell. Number three is cyclic AMP, the second messenger activate one or more protein kinase which may be free in the cytosol or bound to plasma membrane. A protein kinase is an enzyme that phosphorylate aid or phosphorylate group to other cellular proteins such as enzymes. The donor of the phosphate group is ATP, which is converted to ADP. Number four, activated protein kinase phosphorylate one or more cellular proteins. Phosphorylation activate some of these proteins and inactivate others, rather like turning switch on or off. Five, phosphorylated protein in turn cause reaction that produce a physiological response. Uh, Different protein kinase exist within the different target cell and within different organelles of the same target cells. Thus, one protein kinase might trigger glycogen synthesis, a second might cause a breakdown of triglyceride, a third may promote protein synthesis, and so forth. As noted in step 4, phosphorylation by protein kinase can also be inhibited certain proteins. For example, some of these kinase unleashed when epinephrine bind to liver cells and inactivate an enzyme needed for glycogen synthesis. 6. After a brief period An enzyme called phosphodiesterase inhibitor inactivates CEMP. Thus, the cell's response is turned off unless new hormones molecule continue to bind to their receptor in the plasma membrane. Measurement of hormone concentration in the blood. Some most hormones are present in the blood in extremely minute quantity. Some concentration are as low as billion of milligram, 1 picogram per milliliter. Therefore, it was very difficult to measure these concentration by the usual chemical means. An extremely sensitive method, however, was developed about 40 years ago that revolutionized the measurement of hormone, their precursors, and their metabolic end products. This method is called radioaminoassay. Uh, First, an antibody that might specify for the hormones to be measured is produced. Second, a small quantity of this antibody is first mixed with a quantity of fluid from the animal containing the hormone to be measured, and the second is mixed simultaneously with the appropriate amount of purified standard hormone that has been tagged with the radioactive isotope. Uh, Note, uh, there must be too little antibody to find completely both radioactively tagged hormone and the hormone in the fluid to be acid. Therefore, the natural hormone in the assay fluid and the radioactive standard hormone compete for the binding site of the antibody. In the process of computing the quantity of each of the two hormones, the natural and the radioactive, that bind is proportional to its concentration in the assay fluid. Uh, Third, after binding has reached equilibrium, the antibody hormone complex is separated from the remainder of the solution, and the quantity of radioactive hormone bound to this complex is measured by the radioactive containing technique. If the large amount of radioactive hormone has bound with the antibody, it is clear that there was only a small amount of natural hormone to compete with the radioactive hormone, and therefore the concentration of the natural hormone in the assay fluid was normal. Uh, Fourth, to make the assay highly quantitative, the radioimmunoassay procedure is also performed for standard solution of untagged hormone at the several concentration level. Uh, then a standard curve is plotted by comparing the radioactive counts recorded from the unknown assay procedure with the standard curve. One can determine within an error of 10 to 15% the concentration of hormone in the unknown assay fluid. Enzyme linked immunosorbent assay, ELISA. Uh, this method can be used to measure almost any protein, including hormones. This test, this test combines the specificity of antibody with the sensitivity of a simple enzyme is because each molecule of enzyme catalyzes the formation of many thousands of products molecule. Even very small amount of hormone molecules can be detected. Uh, this method is often performed using the plastic plate with the 96 small wheels. Each wheels is coated with the antibody AB1 that is specific for hormone being acid. Fluid containing the hormone is added to each of the wells, followed by the second antibody 2 that is also specific for the hormone but bind to a different site of the hormone molecule. A third antibody is added that recognized AB2 and is coupled to an enzyme that convert a suitable substrate to a product that can be easily detected by colorimetric or fluorescent optical method. Because each molecule of enzyme catalyzes the formation of many thousands of products molecule, even very small amount of hormone molecules can be detected. In contrast to competitive radioamino acid method, ELISA method uses excessive antibodies so that all hormone molecules are captured in antibody hormone complexes. Therefore, the amount of hormone present in the sample is proportional to the amount of product formed. Uh, Here we see that a AB1 and AB2 are antibodies that recognized by the hormone H, they say H hormone, at the different binding sites. And AB3, this one is antibody that recognize AB2E. Uh, E is enzyme linked to AB3 that catalyzes the formation of colored products. P, this one, the red one, from a substrate S with the blue color. The amount of product is measured using optical method and proportional to the amount of hormone in the well. If there are excessive antibodies in the well. Uh, the ELISA method has become widely used in clinical laboratories because it does not employ radioactive isotopes. Second one, much of the assay can be automated using 96 well plates. Number three, it has proved to be cost-effective and accurate method for assessing hormone level. Uh, this was my presentation. Now we want to summarize this presentation that what was the hormone and what that we learned. We want to summarize these things. Hormone are natural organic substance that regulate growth, metabolism, and other function of organ. They are simply biochemical messenger. They can be classified according to the chemical composition organ, whether they work, for example, reproductive hormone and reproductive organ, and if they either act on the small cells producing them, autocrine or paracrine. The multiple hormone system play a key role in regulating almost all body function, including metabolism, growth and development, water and electrolyte balance, reproduction and behavior. And also we learned that a chemical structure and synthesis of hormone, protein and peptides, they're including hormones secreted by the anterior and posterior pituitary gland, steroids secreted by the adrenal cortex, cortical and aldosterone, and the ovaries estrogen and progesterone from testosterone. And we learned that derivatives of the amino acid tyrosine secreted by the thyroid T3 and T4. And also we learned that uh, mm, in general polypeptides were 100 or more that called amino acid called protein, and those with fewer than 100 amino acid that refer to as peptides. We learned that synthesis of these things were in the endoplasmic reticulum of the different endocrine cells. We learned that usually they synthesize that in as a pro-hormones, and then it cleaved to form a prohormone and transport to Golgi apparatus for packing into secretory vesicles, and then these vesicles are stored in the cytoplasm. Then it's expelled as a secretory vesicle fused with the cell membrane, and granular content as extruded. We learned that steroid hormone were usually synthesized from the cholesterol and are not stored in they were not they are stored as the chemical structure of this steroid hormone was similar to that of cholesterol, and in most instances they are synthesized from cholesterol itself. We learned that these steroids are highly lipid soluble, and once they are synthesized, they simply diffuse across cell membrane and enter the interstitial fluid and then to the blood. And we learned that amine hormone are derived from tyrosine. The two groups of hormone derived from the tyrosine, the thyroid and the adrenal medullary hormones, and formed by the action of enzyme inside the plasmic compartment of the glandular cells. We discussed that control of the hormone secretions was most hormone regulatory system work as a negative feedback, but a few works as a positive feedback. Negative feedback prevents overactivity of the hormone system. Feedback regulation of hormone can occur at all levels, including the gene transcription and translation steps involved in the synthetic hormones. We learned also that transport of hormone to the blood, water-soluble hormone peptides and catecholamine were dissolved in plasma and transported from that site of the synthesis to the target tissues. Steroid and thyroid hormone were, in contrast, circulate in the blood, mainly blood-bound plasma proteins, usually less than 10% of the steroid. We discovered that clearance of the hormone from the blood was two factors: the rate of the hormone secretion into the blood and the rate of the removal of the hormone from the blood. It was dependent by these two factors. We learned that hormone are cleared from the plasma in several ways, including metabolic destruction by the tissues and binding with the tissues, excretion by the liver into the bile, and excretion by the kidney into the urine. We learned the principles of the hormone action. Tropic hormone that was primarily for function was regulation of hormone secretion by the another endocrine and synergism activity of the hormone, and permissiveness and antagonism, these activities, and hormone receptors and their activations. We discussed the steps which was involved hormone action to bind a specific receptors, and then on the surface of the cell membrane, and in the cell cytoplasm, and in the cell nucleus. Besides this action of the lipid soluble hormone, free lipid soluble hormone molecule diffused from the blood through intestinal fluid and through the lipid bilayer. We learned that how lipid soluble hormone works, bind to specific receptor in the plasma membrane and form hormone cell receptor complex, which was diffused to the nucleus. Beside this, we discussed how water-soluble hormone works. Water-soluble hormone, the first messenger diffusion from the blood through the interstitial fluid, and then bind to receptors. Adenylate cyclase convert ATP into cyclic AMP, and cyclic AMP, the second messenger, activated one or more protein kinase, may be free in the cytosol, bound to the plasma membrane. And later we discussed that how it measured. For this presentation, these were my references. I hope you listened to me, and thank you from your patience. Inshallah, we will meet in next presentation.